Good day, everyone. I will be discussing the different classifications of hormones, how they are synthesized depending on the type of hormone, and in general, their mechanism of action to certain molecules found in or on the surface of cells or inside the cells, and their effect, okay, once they are bound to those molecules found on a target cell. So this is the outline for today's lecture. And upon completion of this lecture, you are expected to learn all of these. So let's define some terms first. We have endocrine system is comprised of a system of glands that release their secretions directly into the circulatory system. In turn, these glands are actually comprised of cells which are responsible for the synthesis and production of the secretions here, term these secretions hormones. And with a stimulatory signal, these secretions, the hormones, are actually secreted into the circulatory system. And from there, a lot could happen. So it may be bound to the transport protein and is brought to its target tissue. Or if it's water soluble, that hormone could bind directly to the surface molecules, okay, and generate an effect on that particular tissue. So we have another term, endocrinology. It's the study of hormones and disorders associated with abnormalities of these hormones. Half-life is the time required for a hormone to fall to have its original concentration in the circulation or other specified body fluid. And a hormone is a chemical substance that has a specific regulatory effect on the activity of a certain organ or pituitary gland. Then we have the hypothalamo-hypophyseal system. So I will be discussing more of these on the second part of my lecture. So it's comprised of a system of neurons, fiber tracts, endocrine tissue, and blood vessels that are responsible for the production and release of pituitary hormones into the systemic circulation. So bear in mind that this hypothalamo-hypophyseal system is actually comprised of so many, okay, so many things: neuron, fiber tracts, endocrine tissue, and blood vessels. They in turn, okay, they produce hormones and facilitate transport of these hormones to a particular organ and eventually into the systemic circulation. Then we have this term receptor. It's a molecular structure within a cell or on the surface characterized by selective binding of a specific substance, a specific physiological effect that accompanies the binding. Now I have to put emphasis on the phrase here, is a structure within a cell or on the surface of the cell, because receptors, hormone receptors, may actually be found inside the cell or on the surface of the cells. Now, there are receptors, say for example, receptors for thyroid hormones, which are actually found on, on the nucleus. Okay? So within the cell, it may be found in the cytoplasm or in the nucleus. Say for example, receptors for thyroid hormones, or they may be found on the surface, okay, on the surface of the target cell. Now, upon binding of the hormone to the receptors found within a cell or on the surface of the cell, they generate a specific physiological effect. I will be discussing this in detail in the next few slides. Now, I have mentioned that the endocrine system is actually comprised of a system of glands. And these system of glands are actually responsible for the production of a particular hormone. Say, for example, your hypothalamus produces releasing hormones. For example, we have corticotropin releasing hormone, growth hormone releasing hormone, thyroid releasing hormone, and among others. Now, these releasing hormones act upon the cells in the pituitary gland to produce another set of hormones. Say, for example, your thyrotropin releasing hormone acts on thyrotropes, or the cells responsible for production of another hormone we call TSH, thyroid stimulating hormone. Another example are your CRH, or your corticotropin releasing hormone. They act on corticotropes, okay, found in the pituitary gland to produce another set of hormones. Now, I will have a more detailed discussion on these hormones, okay? Each specific hormone produced by each specific gland on my next lecture. So please keep posted for that lecture video. Now, in turn, hormones produced from the pituitary gland when secreted in the circulatory system, okay, they are brought to the specific target tissues. Let's say, for example, your TSH, okay? So your TSH, your thyroid stimulating hormone, acts upon the cells in the thyroid gland to produce thyroid hormones, particularly T3 and T4, okay? And among others, okay? A lot, no? So what I'm trying to point out here is that your endocrine system actually is comprised of a system of glands, a lot of glands. And these glands produce a specific hormone which is responsible for a certain physiological effect. Now, the word hormone is derived from a Greek term that means to arouse activity. So, as classically defined, a hormone is a substance that is synthesized in one organ and transported by the circulatory system to act on another tissue. However, this original description is too restrictive because hormones actually can act on adjacent cells. So we call when hormones act on adjacent cells or neighboring cells, we call that paracrine action of that particular hormone. And on the cell, and if they act on the cell in which they were synthesized, we call that autocrine action, okay? Without even entering the systemic circulation. So hormones has a hormone has a specific regulatory effect on the activity of an organ or organs. So you learn more of these, no, these regulatory effects certain organ as we go on with our discussion. So hormones can be classified according to its chemical composition, its solubility properties, the location of receptors, and the nature of the signal used to mediate hormonal action within the cell. So let's start first with its classification according to chemical composition. It may be a peptide and a protein. Okay? So generally, peptide and protein hormones are water soluble, and when secreted into the circulation, being water soluble, they are not bound to any carrier protein. Now, its half-life in plasma is 10 to 30 minutes. So this is actually shorter, okay, shorter half-life compared to other types of hormones, say for example, steroid hormones, which are actually bound to a carrier protein. Now, being not bound to a carrier protein, they have shorter half-life in the plasma. So it may range from 10 to 30 minutes. So they are synthesized and stored within the cell in the form of secretory granules. Let me expound that using this figure here. Now, originally, you learned that proteins are synthesized in the endoplasmic reticulum, okay, with a proper signal directed by the nucleus, the information copied from the nucleus. These material, okay, are actually translated into proteins in the endoplasmic reticulum. In other words, what I'm trying to point out is that synthesis of these peptide and protein hormones occur or happens in the endoplasmic reticulum. Now, okay, initially they are synthesized as a large molecule without any biological effect. These large molecules are what we call pre-pro hormones. So pre-pro hormones are synthesized in the endoplasmic reticulum. Now pre-pro hormones are modified, okay? So a portion of that hormone is cleaved, okay, producing a smaller molecule we call now prohormone. Now this prohormone is transported to the Golgi apparatus and is packaged, okay, packaged into secretory vesicles. Again, prohormones from the endoplasmic reticulum are transported into the Golgi apparatus for packaging into secretory vesicles. Now, inside that secretory vesicles are actually enzymes. Now, these enzymes in turn would, okay, would cause cleavage of the prohormone producing a biologically active hormone molecule plus the inactive fragment which was removed from the prohormone. Okay? So again, the prohormone itself is actually comprised of an inactive fragment and the hormone itself. But in the Golgi apparatus, it is packaged, okay? It is packaged into secretory vesicles, and inside that secretory vesicles are enzymes which degrade, okay, which cleaves the prohormone into smaller hormones. Two molecules are produced upon that cleavage, the inactive fragment and the hormone with the biological activity. Now, with a stimulatory signal, okay, with a stimulus, what happens is that these secretory vesicles, okay, travels into the cytoplasm and eventually binds, okay, it binds to the cell membrane and extruding via the process of exocytosis or secreting the contents, no, of the different vesicles, no? Secreting the contents of the vesicles via exocytosis into the extracellular fluid. And from there, the hormones may be bound to a transport protein or it may be brought, okay? It acts directly on to the target tissue being water soluble. Now, peptides and proteins being water soluble cannot cross the cell membrane. So you might ask, if they can't cross the cell membrane, how do they initiate, okay? How do they produce the physiological effect that I have been talking about? So when the hormone is released from a particular gland, no, or cell producing it, peptides and protein hormones, since being not able to cross the phosphobilipid layer, being water soluble, could not cross the phosphobilipid layer of the cell membrane. And so it generates its effect by binding to another molecule that is embedded on the surface of the cell membrane. And we call this molecule now receptors. Okay? So a peptide and a protein hormone, for it to generate a physiological effect, it binds, being a water-soluble hormone, and could not pass through the membrane. It binds to cell surface molecules, okay, which are embedded in the surface of the cell. So I will discuss this in the next few slides in detail. So another classification of hormone as to chemical composition are steroids, steroid hormones. They are lipid molecules that have cholesterol as a common precursor. Okay? That cholesterol, being a common precursor of steroid hormones, is comprised of three cyclohexyl rings, 1, 2, 3, and one cyclopentyl ring. 3 cyclohexyl ring, 1 cyclopentyl ring. They are produced by adrenal glands. So the hormones produced, the steroid hormones produced by the adrenal glands are cortisol and aldosterone by the ovaries. For example, estrogen and progesterone by the testes, testosterone, and the placenta, estrogen and progesterone. So what they are water insoluble, okay, being made from cholesterol, they are hydrophobic or water insoluble. And so they, in the plasma, in the circulatory system, they circulate bound to a carrier protein. Okay? Now, being water insoluble, they enter the cell easily by passive diffusion. Since, remember that a cell has a phosphobilipid layer. And being water insoluble, being comprised of cholesterol, they actually diffuse easily into the phosphobilipid layer of the target tissue. Okay? So for example here, a lipid lipophilic hormone or a steroid hormone being water insoluble and comprised of cholesterol precursor molecule, it diffuses easily into the cell surface membrane. And then from there, the receptors of steroid hormones are found in the cytoplasm, or some are found in the nucleus. Okay, so from there it may bind to its nucleus. So it may bind to its receptor in the nucleus or in the cytoplasm rather. So being bound to a carrier protein, it has a longer half-life compared with steroid hormones. So its half-life is 30 to 90 minutes, depending on the type of hormone, because some hormones actually have longer half-life, may range to days, even days. Then we have amines or amino acid related hormone. So from the term itself, no, they are derived from amino acid, and they are intermediary between steroid and protein hormones. So they are water soluble and they circulate in the plasma either bound to proteins, or they may circulate unbound, not bound proteins. They are freely circulating. So they may be bound to proteins or they may circulate freely in the plasma, not bound to any proteins. So there are different physiological actions of hormones depending on the type of hormone. So we have hormones responsible for growth and development. Okay, so we have your gonadotropins. So we'll discuss each effect of the hormone in my succeeding lecture videos. So what I'm trying to point out here is that the hormones actually have different physiological effects. Now, it may cause growth, okay? It may facilitate growth and development. Say, for example, your gonadotropins, which is responsible for development of your primary and secondary sexual characteristics. Okay? Your ACTH, okay, pr- produces hormone, and also your TSH, they produce hormones, which actually has a role in metabolism. Now, hormones, okay, also exert homeostatic control of metabolic pathways. For example, regulation of blood glucose. So what hormone is this? We have insulin, okay? We also have glucagon, or metabolism or homeostasis of calcium. What hormone? Parathyroid, okay? And then water and electrolytes. So water may be ADH, electrolytes may be aldosterone, and among others. No? So you will have a separate discussion on those specific hormones. They also facilitate regulation of energy production, use, and storage. I've mentioned that some hormones actually play a role in metabolism, and in turn they regulate, okay, energy production and storage. So the unique or specific action of a hormone on its target tissue is a function of the interaction between the hormone and its receptor. So I've mentioned before that for a particular hormone to generate a physiological effect, it has to be bound first to its receptors. And a receptor may be found on the surface of the cells or inside the cell, no, intracellularly. Maybe could be in the cytoplasm or could be in the nucleus of the cell. So the concept of the target cell is a useful way of looking at the hormone action. So before, it was thought that hormones affected a single type or only a few kinds of cells, and that a hormone elicited a unique biochemical or physiologic action. We now know that a given hormone can affect several different cell types, and that more than one hormone can affect a given cell type, and that hormones can exert many different effects in one cell or in different cells. Now I want you to look at the diagram here. Now this diagram actually summarizes the concept of the target cell, no? The target cell theory. Now the substances that you see here, okay, in the extracellular fluid are different molecules, no, circulating in the extracellular fluid. But of these substances circulating in the extracellular fluid, only a few are recognized by hormone receptors. Now, the boxes here labeled 1, 2, 3, 4, 5, 6 are the target cells, okay? The cell types to which hormones bind to. Now, a cell, okay? A cell may contain receptors, okay, here, or it may not contain any receptors. Now, receptors found in the cells must select these molecules from among high concentrations of other molecules found in the extracellular fluid. Now, this simplified drawing shows that a cell may have no hormone receptors. No? Particularly cell number 1 doesn't have any receptor to any hormones for any hormones. Or a cell may have one receptor for a particular hormone. Say, for example, cells 2, 5, and 6. Or it may have a cell, no? A particular cell may have a receptor for several hormones. Say, for example, cell number 3. Or a cell may have a receptor, but no hormone is found in the vicinity. So no one will bind to this receptor since no hormone is found in the vicinity. So with the discovery of specific cell surface and intracellular hormone receptors, the definition of a target has been expanded to include any cell. Okay? So by the concept of the target cell theory, a target is any cell in which the hormone binds to its receptor, whether or not a biochemical or physiologic response has yet been determined. Okay? So to put it simply, any cell which has a receptor for a particular hormone is called a target or a target cell. So let's discuss first these surface receptors. So these cell surface molecules actually are embedded on the surface of the cell membrane, spanning, okay, typically spanning 7 times the cell membrane. So they have 7 alpha helical membrane spanning domains shown here. So 1, 2, 3, 4, 5, 6, 7. 7 domains, of which the amino terminal is actually found extracellularly and the carboxy terminal intracellularly. Now, these cell surface molecules, receptors, are coupled to another complex of proteins. No? We call it the G protein complex. So it's a complex because it is comprised of 3 subunits, the alpha, the beta, and the gamma subunits. The beta gamma subunit, okay, is anchored to the plasma membrane through its prenylated groups. And the alpha subunit is anchored to the membrane through its meristölated groups. Now, in its inactive form, the G protein is bound to GDP or guanosine diphosphate. And when a hormone is bound to the surface of the receptor, causing a conformational or configurational change, okay, on the orientation of the receptor. This causes now GDP to be displaced and replaced with GTP. In turn, the alpha subunit dissociates from the G protein complex and it causes the activation of the effector cell molecule. Then we have your intracellular receptors. So I've mentioned that your intracellular receptors are actually found maybe in the cytoplasm or could be in the nucleus, okay? Depending on the type of hormone that binds to it. Now, once a steroid hormone passively diffuses in the cell surface membrane, okay? So it binds into a cytoplasmic receptor, forming a hormone receptor complex. Now, this hormone receptor complex is in turn the effector molecule, causing now the physiological effect of that particular hormone. Or it may bind to its receptor on the nucleus, okay, on the nucleus, forming now a hormone receptor complex which serves as the second messenger or the effector molecule, causing now it changes, okay, or resulting to a physiological effect. Now, a receptor in general is comprised of many domains, okay? So, it has a ligand binding domain. This is the particular portion of the receptor which binds to the hormone, okay? Hormone is also termed as ligand. So thus, the term ligand binding domain. So this is where the hormone binds to. Now it has activation domains. Now remember, once the hormone is bound to the ligand binding domain, it the receptor, okay, the hormone causes the receptor to undergo configurational changes, thus exposing its activating domains. The activating domains in turn acts upon the effector molecules to produce the desired physiological effect. Now, for steroid hormones, there is okay, an additional part of the receptor we call the DNA binding domain. So you do not see this in surface receptor, cell surface receptor molecules, but you only see the DNA binding domain in cytoplasmic receptors and nuclear receptors, since these receptors are the only receptors which goes inside the cell to produce a certain physiological effect. Now, each of the different hormones has its own characteristic onset and duration of action, meaning the hormones, the effect of the hormone, the duration of the effect of that particular hormone actually varies depending to the type of hormone. Say, for example, amino, peptide, and protein hormones actually okay, exerts its action in a shorter period of time, having a shorter half-life. Then, on the other hand, steroid hormones being bound to transport proteins, okay, so they actually exert longer duration of action, having a longer half-life. So highly specialized mechanisms are available in the target tissue that allow minute quantities of the hormones to exert powerful control over the physiological systems. Okay? And I will be discussing these specialized mechanisms in the next few slides. So hormone secretion is controlled by feedback. We term it as negative feedback if we want to okay, prevent further secretion of the hormone. We term it as positive feedback if we want more secretion, more production of that particular hormone. And hormone release actually occur in cyclical variations, meaning depending on the time of the day, the season, the age of the patient, the hormone release is affected by a okay, particular time of the day. So for example, cortisol. Okay? Cortisol is a glucocorticoid, a hormone produced by the adrenal gland. Now, there is actually a particular time wherein you reach a peak or high levels of your cortisol, and that is preferably after waking up or in the morning. So cyclical variations. Okay? So this refers to the diurnal rhythm, diurnal variations of which the hormone is released, affected by age, the time of the day, the seasons. So transport of hormones in the blood. So I've mentioned that water-soluble hormones being water-soluble, being hydrophilic, okay, they actually are not bound to a okay, certain transport proteins. Now, with exception to amine hormones, because some amine hormones may be bound to transport proteins. Now, water-insoluble hormones being lipophilic in the circulation, they are bound, okay? So they are bound to transport proteins. Now, hormones are cleared from the blood via metabolic destruction, okay, via degradation of the hormone by the target tissue or by binding with the tissues, okay, or by excretion in the liver through the bile or excretion in the urine. Now, I've mentioned that there are actually there are actually receptors, okay, in a target cell. And once the hormone is bound to that particular receptor, it generates physiological effect. Now, that mechanism of activation of these hormones actually would vary depending on its location. Okay? So say for example, let's start with cell surface receptors. So once a hormone in the vicinity binds to its particular receptor in the cell surface, okay? So remember your cell surface receptors are coupled to a complex of protein we call G protein. And why do we call this a complex? It's because this is actually comprised of three subunits, the alpha, the beta, and the gamma subunits, of which the beta gamma subunits are embedded, okay, or are attached, are secured to the cell surface membrane, the cell membrane via its granulated subgroups, and the alpha subunit via its merstoylated subgroup. Now, in turn, the alpha subunit is, we learned from the previous slides, it is actually bound to a GDP, guanosine diphosphate molecule, thus the term G protein. Now, the receptors to which these G proteins are coupled into are called GPCR or G protein, okay? GPCR, G protein coupled receptors, meaning that particular receptor is attached to or coupled to a G protein. Now, once the hormone is attached to the cell surface receptor, it causes a configurational changes on that particular hormone. Now, that configurational changes causes the GDP of the alpha subunit in the G protein to be exchanged with a GTP. Okay? So GDP, once the hormone is bound now, the hormone receptor complex undergoes, the receptor undergoes configurational changes, and the GDP is exchanged with a GTP molecule in the alpha subunit. And once that happens, the alpha subunit dissociates itself from the beta and the gamma subunits, and in turn, the alpha subunit acts upon, it may act upon an enzyme or a target protein, which is the effector molecule or the second messenger. That in turn, the second messenger will actually cause the physiologic effect of that particular hormone. So for example, so your GTP, okay, being bound to the alpha subunit may act upon to the effector molecule. What is that effector molecule? It may be an enzyme. So in turn, no, in this diagram here, it may act upon an enzyme. So the enzyme here is adenylyl cyclase. So adenylyl cyclase now catalyzes conversion of ATP to CAMP. So cyclic, okay, adenosine monophosphate. So the CAMP now itself is the second messenger, which generates the physiological effect, okay, which generates the cell response. How does it generate cell response? So it may cause increase, it may in turn affect, no, intracellular ion. So the CAMP in turn, okay, would actually generate the desired physiological effect of that particular hormone, no, to generate a cell response. So again, what I want to point out here is that the alpha subunit may act upon, okay, it may act upon the effector molecule. And that effector molecule may be an enzyme, it may be an ion channel, okay, it may be an enzyme or an ion channel, or it may be a protein, which generates a second messenger. So adenylyl cyclase generates the second messenger CAMP. The second messenger in turn will generate the physiological, will cause, no, will cause the physiological effect of that particular hormone, thus producing a cellular response. Now, another, okay, another mechanism is that instead of adenylyl cyclase, the alpha subunit may act upon another enzyme. So that enzyme may be phospholipase C. Now, your phospholipase C generates another set of okay, second messenger system. For example, your IP3, your inositol phosphate 3, the second messenger here. And in turn, your second messenger will generate, will generate the desired physiological effect. Now, I will not be discussing the physiological effect because you will actually have a separate lecture for each hormone because each hormone will have a different physiological effect. What I want to establish here are the different mechanisms on which these physiological effects may be generated. So it may be generated by the action of the alpha subunit on effector molecules, maybe an enzyme. So maybe adenylyl cyclase or maybe phospholipase C or maybe another set of target proteins. Now, it is actually different for intracellular receptors. Okay? So, by the way, by post-receptor action, I'm trying here to establish what happens after the hormones binds to the receptor. So, thus, the term post-receptor action. So, it's actually a different set of events that happens with intracellular receptors. So, using the same diagram. Now, I've mentioned that steroid hormones actually being the derivative of a cholesterol precursor molecule can diffuse easily. They diffuse passively into the cell membrane, which is comprised of a biphospholipid layer, bilipid layer. And so from there, it may be bound to a cytoplasmic receptor or a nuclear receptor. So your intracellular receptors may be found in the cytoplasm, so we call it cytoplasmic receptor, or it may be found inside the nucleus, so we call it a nuclear receptor. Now, once bound to the receptor, it produces a complex, a hormone receptor complex. And in contrast to the cell surface molecule receptors producing second messenger, it may be CAMP or IP3, for steroid hormones, the second messenger which will generate the desired physiological effect is the hormone receptor complex. Okay? So, when it binds to a cytoplasmic receptor forming a hormone receptor complex, this travels, it goes inside the nucleus. In turn, the hormone receptor complex binds to a particular portion of the DNA we call the hormone response element. Now, this, upon binding of the hormone receptor complex with the hormone response element in the DNA, it generates now the desired physiological effect. Now, if the receptor is found in the nucleus, so the hormone goes directly inside the nucleus. So, for example, this is common for thyroid hormones. So it goes directly inside its receptor in the nucleus, and the hormone receptor complex is the second messenger now, which when bound to hormone response element generates the physiological effect of that particular hormone. So again, I will not discuss the physiological effect of that hormone because you will have a separate lecture for each physiological effect for each hormones. Now, okay, so this is actually an example of how the thyroid hormone generates its physiological effect. So thyroid hormone receptors, as I've mentioned, are actually found inside the nucleus. And these thyroid hormone receptors, once bound to the hormone itself forms a complex we call the thyroid receptor complex, the thyroid hormone receptor complex, or the hormone receptor complex. And the hormone receptor complex binds to a hormone response element. So we call that hormone response element, no, acting on the thyroid hormone receptor complex as the thyroid receptor element. Okay, the hormone receptor element, thus causing now or generating its physiological. Now that ends my discussion. Please keep posted for the second part of my lecture. Thank you very much.